This is part 4 of the Chapter 10 lecture notes, and I wanted to go back to this slide where we were discussing negative regulatory molecules. The RB, or retinoblastoma protein, when in its normal state, it is bound to E2F, which is a transcription factor. And as long as it's bound, it keeps the cell cycle from continuing. But if the cell grows as it is supposed to do, if it grows in a predictable manner and it's large enough, the size is large enough, then the presence of ATP will cause the RB protein to become phosphorylated, and it will disconnect from E2F, and then E2F will bind to DNA, and it will cause DNA to continue that cell cycle, to advance the cell cycle. So it's a negative regulator because it's most of the time it keeps the cell cycle from continuing. It prevents the cell cycle from continuing. Now, how is cancer related to the cell cycle? Cancer is basically uncontrolled cell growth. So we just talked about how the cell cycle is regulated, and we don't want the cells to grow out of control, because what happens then is we get a tumor. So, and we've been, maybe multiple tumors. So what starts as a gene mutation in a gene results in a faulty protein that regulates cell reproduction. So one of those positive or negative cell cycle regulators, when it is damaged because of a gene mutation, this can cause cancer. So tumors result when these mutated cells begin to grow at a rate that surpasses the growth rate of normal cells. Now, proto-oncogenes are normal genes, and if they are mutated, they can become oncogenes, and oncogenes are cancerous genes. So proto-oncogenes code for positive cell cycle regulators, and one of those is P53. P53 is a positive cell cycle regulator. So in the normal situation, DNA damage in the cell cycle, DNA damage or cell cycle abnormalities or hypoxia, that is low oxygen, any of these conditions, P53 normally will lead to either programmed cell death called apoptosis, or it will cause the cell cycle to stop, the DNA to be repaired, and the cell cycle to start over. Or if the DNA can't be repaired, then apoptosis will result. So P53 can lead to programmed cell death or apoptosis when that cell is could possibly become a cancerous cell. But if P53 is mutated and DNA damage is detected, or cell cycle abnormalities or low oxygen hypoxia, it allows the cell cycle to continue, and the cell can actually become cancerous. The cell cycle will continue even when these things are detected. There are genes called tumor suppressor genes, and these are genes or segments of DNA that code for negative regulator proteins. So, for example, like RB, retinoblastoma protein. So when these genes are activated, they can prevent uncontrolled division. They can suppress the growth of tumors. Cells with a mutated form of a negative regulator might not be able to stop the cell cycle if there is a problem. So tumor suppressor genes are good. We need those genes because they prevent uncontrolled cell division. And Okay, so moving on, we really have focused on the cell division of eukaryotic cells, but prokaryotic cells divide as well, and cell division in prokaryotic cells, such as bacteria, is called binary fission. Binary means two, fission means split, so that word literally means to split in two. And this is how that process works. Binary fission in prokaryotes works by the parent cell, the original prokaryotic cell, remember, has a single circular chromosome, and it also contains proteins called FTSZ proteins. And these FTSZ proteins migrate toward the midpoint of the cell, and they lead to the duplication of that single circular chromosome into two circular chromosomes. And then basically those FTSZ proteins will form a ring that will cause the cell to split in two, and form not only that, but form a septum or a divider between the two cells. And eventually the original parent cell pinches off and forms two new prokaryotic cells.